In this lecture, we are going to start our study of acids and bases. Now, you've learned about acids and bases before, so just a quick reminder, we have our pH scale here, roughly runs from 0 to 14. When we look at acids and bases, acids are on the lower end of the scale, so they typically have a pH below 7. The more acidic the solution, the lower the pH. Bases are alkaline. They have a higher pH number. The higher the pH, the more alkaline the solution. And a pH of 7 is neutral, which means that that solution has the same number of hydrogen as hydroxide ions. When we look at pH, we're looking at these protons. H plus is called a proton, and acids always donate hydrogens. So when you add an acid to a solution, you're going to increase the number of hydrogen ions. And when you add hydrogen ions to a solution, it makes the pH number go down. So it's an inverse relationship. The higher the hydrogen ion concentration, the lower the pH. You might remember it's a log scale. So every change in number is a 10-fold change in hydrogen ions. So going from 7 to 6 means that a pH of 6 solution has 10 times more hydrogen ions than one of 7. But a pH solution of 5 has 100 times more hydrogen ions than the pH solution that was 7. The base, on the other hand, are substances that release these hydroxyls or hydroxides. And so if you have an alkaline solution, more of those hydroxides are released. And what they do, though, is they take the hydrogen. They actually form water. So when you add a base to a solution, it will donate hydroxyls, but what it actually does is it lowers the hydrogen ion concentration. So the lower the hydrogen ion concentration, the higher the pH value. We will be looking at weak and strong acids and bases. A weak acid or base is something that when you add it to a solution, it does not completely dissociate, which means it does not have a significant effect on the pH of the solution. A strong base or strong acid will completely dissociate, which means it will completely split apart and have a much greater effect on the solution. Our weak acid is carbonic acid. Hopefully you recognize that from respiratory. And our strong acid that we'll be using as an example is hydrochloric acid. Our strong base, this is sodium hydroxide, and our weak base will be bicarbonate ion. So as I mentioned, when we add these acids and bases to solutions, the difference is that a strong acid or base will completely dissociate. So if we were to add a strong acid to solution, notice that it completely separates into hydrogens and chlorides. So that's really going to drop the pH because it's the free hydrogen ions that we worry about. Whereas carbonic acid does not completely dissociate. A couple do, but mostly it doesn't. And that's true for the base as well. So when we teach this, or when I teach it, I like to just say strong acid or strong base, bad. Weak acid or weak base, weak acid or weak base, good, because they won't affect pH as much. We know we have a range of pH, and we just looked at this. When we look at arterial blood, so I'm talking about arterial blood, the normal range is pretty narrow, 7.35 to 7.45. In fact, if you look at human physiology, our blood pH is typically between 7 and 7.8. And once we go beyond those, death typically is going to occur because we just can't survive. So it's a pretty narrow window. When we're looking at pH, anything below 7.35 is acidosis. If you were in a chemistry class and you said that 7.30 was an acid, you'd probably be marked incorrect. But in a human, it is. So we call it physiological acidosis because it's acidic to us. Alkalosis would be anything greater than 7.45 for your arterial blood pH. You will most likely see acidosis more than alkalosis. However, both are dangerous, as you saw on the previous slide. Both can cause death. It's just that the body makes a lot more acids than bases, and so you're more likely to see the acidosis when you're in healthcare rather than alkalosis, although it does exist and you might see it. When we're looking at signs and symptoms, just know generally our brain, our nervous system is pretty susceptible 2, these pH ranges. Notice 2 alkaline, 2 acidic can cause a coma to occur. We also worry about the cardiovascular system, and you can see that it can affect different systems in the body, and both are not desirable. When we look at acids, acids usually come from 2 places: what we eat and generate through metabolism. You might remember we have pyruvic acid, keto acids, and then, as I mentioned, what we eat. So these are the main source of these acids. When we look at acids, they can be characterized as volatile, and volatile just means that basically think of them as you can get rid of them in your lungs. They can change into a gas and be eliminated. And so when we look at the volatile acids, we're really going to be looking at carbonic acid. This equation should look very familiar to you. This is also one of the most important equations for buffering. And we're really looking at CO2 levels. We know that PCO2 is one of the most important factors. We knew our respiratory system was normally able to deal with all that CO2 levels. But if we were to increase our CO2 levels, like maybe you had emphysema or pneumonia, then what would happen is this equation would go to the right, and notice you're going to generate more hydrogen ions. So if my CO2 levels increase beyond normal, then notice I'm going to increase my hydrogen ion concentration because I'll make more, which means that my pH will fall. And so when we look at respiratory, we really watch the CO2 because the CO2 is pretty important at predicting the pH. Now, the lungs can be the cause of the problem, as they are here. You'll see later we'll talk about things that are called respiratory acidosis, when the lungs, the CO2 isn't being eliminated, and we're having an acidosis occur. But also, if you have healthy lungs, if the problem's somewhere else in the body, the lungs can also fix it. So this equation will be looking at how when you have an abnormal condition with your CO2, you might generate more hydrogen ions. Or if the CO2 is fine, we'll be looking at the other end, how it helps buffer if there's too many hydrogen ions. So you're more likely to see respiratory acidosis. You could see an alkalosis occur if maybe you were hyperventilating, and you had too little CO2. Then you always want to maintain your CO2 ranges. You might remember the range that you learned previously was 35 to 45 was my normal PCO2. So if I'm getting rid of it, then the equation would go the opposite direction, and now I could have an alkalosis, but we're going to see more acidosis. So then we also have our fixed or organic. These are a lot of the ones that are either from our diet or produced from metabolism. And so we have lactic acid, pyruvic acid, keto acids, just a whole bunch are always generated. So every day we generate acids and we have to get rid of them and buffer them. 3 ways we buffer. Chemical are fast, seconds to minutes. So these operate first, your first line of defense, they jump in immediately. But they're limited. You can use up these chemical buffers. These are the chemical buffers we'll be looking at. Once they fail, if you've overwhelmed them, you go to your respiratory. As I mentioned, our lungs are very powerful buffers as well. Now, these usually take minutes to hours to buffer. And if they can't do the job, we have our kidneys. And our kidneys directly eliminate hydrogen and bicarb in our urine. So these are the slowest to kick in, but the longest lasting, the more complete buffer, because you actually eliminate the hydrogen and the bicarb in the urine. In today's lecture, we're going to focus on the chemical buffers. And the chemical buffers usually exist in pairs. And what they're going to do is either donate a hydrogen or remove it. We know that pH is due to the free hydrogen ions. And so we can alter those hydrogen ions, we can fix the pH problem. So we'll be using weak acids and weak bases. There's always a pair. So a buffer pair has a weak acid and a weak base. The acid will donate the hydrogen. So if your pH level has gone up, remember the reason it's up is that you don't have enough hydrogens. There's too few. So now you want the acid to donate hydrogens to bring them back to normal. Or if my pH is falling, we know that if it's falling, it's because I had too many hydrogens. Now I want to get rid of them. So the base will bind to it. And so what they do is they can pick up or release hydrogen ions to control the pH. So when the pH falls, as we mentioned, when the pH decreases, we have too many hydrogens, so we want to eliminate them. And if the pH goes up, we don't have enough hydrogen, so now we want our acid involved because it will donate or release hydrogen to bring it back down. When we look at the chemical buffers, we'll be looking at three. We have the protein buffer systems. We have two chemical buffers we're learning. The sodium bicarb, which is carbonic acid bicarb, and phosphate. If we look at the proteins first, there's two pieces to the protein buffers. The first one to know is that every amino acid can work like a buffer. Amino acids always have an acid group, the COOH was a carboxyl group, and they also always have this NH2 group. That's why they're called an amino acid. So in Bio 119, you would have learned that there are the two pieces, and then we always just say R is the rest of the molecule. So this acid group can release a hydrogen and work like a weak acid, and this NH2 group can actually pick up a hydrogen to form ammonia and work as a base. So it's pretty cool that the same molecule can either donate hydrogens if the pH is too alkaline, or remove them if the pH is too acidic. The other buffer you learned for protein is back on hemoglobin. Hemoglobin was a protein and it could transport O2 and CO2. The unbinded, if that's a word, form, remember, it always has a little bit on it, but we had reduced hemoglobin, and then we had carbamino hemoglobin and oxyhemoglobin. So anytime there's a hydrogen hanging out, hemoglobin actually wants to buffer. It actually grabs that hydrogen, and when it does it, it's going to drop off either the CO2 or the O2. So it's actually buffering, and it works out perfectly for us because when that happens, it allows it to unload the oxygen or unload the CO2. And you saw how the processes were linked and combined through the Haldane effect to allow this to happen. So hemoglobin is a great buffer. In fact, anytime our pH fell, when we had too many hydrogen ions, you might remember we increased the O2 unloading. We let go of more oxygen to our cells, which was perfect. Now let's look at these chemical buffers. We're going to look at the sodium bicarb first, and this is a buffer that happens in your extracellular fluid and your intracellular fluid. Happens a lot in your blood. And when we're looking at our buffering, what you're seeing in this picture is basically a solution of water. It's neutral because it has about the same number of hydrogens as hydroxyls. Notice if we were to add an acid to the solution, we would basically drop our pH because we're releasing all these hydrogens. It would split apart and we'd notice the hydrogens increases, so the pH falls. Now we had a buffer. What the buffer does is it helps to bind to some of those free hydrogens. Notice there's fewer of the free ones down here. The bicarb buffer is going to grab the hydrogen and form carbonic acid. So notice now the pH is very slightly changed because I had my buffer. The same thing happens with the base. If I were to add the base, I have all these hydroxyls. Notice now there's fewer hydrogen ions. Well, if I have the buffer, now what happens is my buffer is going to prevent all those hydrogens from being removed. It won't release as many of these hydroxides to cause a neutral pH change. So the buffer is important because it helps prevent drastic pH changes. And when we look at this buffer, sometimes it's called sodium bicarb because a lot of times you'll see a little sodium added to the bicarb. It's very similar to what we learned, except now you have a sodium, because sodium bicarb is how it likes to exist in your ECF, because sodium is one of the more numerous cations out there. And so we're going to have my weak acid is carbonic acid, and my weak base is my sodium bicarb. So I always have a buffer pair, a weak acid and a weak base. And so the way it works when we're buffering is, remember, strong acids, bad, weak acids, good. So how do we buffer? Well, if you're buffering an acid, here's my strong acid, my HCl. Now my base gets involved. It combines with the strong acid, and notice it converts the strong acid, HCl, into carbonic acid, H2CO3. This will not change the pH as much. And then I also form a salt. An easy way to look at it is to think about the sodium, I'll put that in purple, and the hydrogen, I'll put blue. The hydrogen and the sodium will swap places in all of these equations. And so if my hydrogen comes over, I would have H2CO3. And if my sodium swaps, notice the sodium comes over here, so I have NaCl. That's all it is an exchange reaction. But what I do is I get rid of my strong acid, and now I have a weak acid, so it won't change the pH. Very similar thing when we're doing the base. So if I have a strong base, I need to get rid of the strong base and replace it with a weak base. So if I have a strong base on the left side of my arrow, I need to have the weak one on the other side. So now my acid gets involved. Well, what's going to happen now? Same thing. My sodium and my hydrogen are going to swap places. So technically I would have HOH. There's my hydrogen, the water. And then remember my sodium swapped. There's my sodium. Sorry, got a little messy. So my sodium comes over. I have Na, sodium bicarb, and water. So when I buffer a strong acid, I get a weak acid and a salt. And when I buffer a strong base, I get a weak base and a water. And this is the sodium bicarb buffer. Understand that inside a cell, it's the exact same equation, but instead of sodium, you would have either potassium or magnesium because there's very little sodium inside the cell. And this is a real powerful buffer, but it's dependent on how much of the buffer you have. How much of the sodium bicarb do you have? And we know we have to have a certain ratio of this for it to function properly. We have to have a base bicarb to carbonic acid ratio of 20:1. So for every carbonic acid molecule, we need to have 20 bicarbs for this system to work properly. If we start to run out of them, we're going to have an issue. So if we were to drop our ratio, so maybe it was 10:1, now I only have 10 bases for every acid. Well, I'm going to expect this when I'm in an acidosis state. That's not going to be good because I need 20 of them. Or if for some reason I start holding on to bicarb, now I have too much base. This is going to happen and I'm going to notice an increase in pH. So we like to look at the bicarb levels to also help us figure out what's happening in pH. And when we look at the bicarb levels and see the effect on pH, we call these things metabolic acidosis or metabolic alkalosis. The phosphate buffer is the last one. You're just memorizing the chemistry. You have to have an acid and a base. Notice the acid has 2 hydrogens and the base has 1. That's how I remember them. Can't do it for all acids, but it works for this. So this is just sodium dihydrogen phosphate, and this is sodium monohydrogen phosphate. You never have to say it, but notice same thing. If I have a strong acid, the goal is to get rid of the strong acid and replace it with the weak acid. So now my base gets involved, and I'm going to have the same thing. My sodium, actually I think I used blue for hydrogen, so my hydrogen and my sodium are going to swap places. So my sodium comes over to the salt and my water comes right over here. Excuse me, my hydrogen, excuse me. So my hydrogen. So the hydrogen comes over. So sodium and hydrogen swap places, same equation. Same thing will happen for the base. Now I have a strong base and a weak acid. The goal, strong base bad, weak base good. So I have the strong base on the left. I better have the weak base on the right of the arrow. That's another check. If I have the strong acid on the left side of the arrow, I have to end up with the weak acid on the right side of the arrow. So now I'm buffering the strong base. The acid gets involved. And now what I'm going to do is sodium and hydrogen are going to swap places. So hydrogen comes over here. So I have my H2O and my sodium is going to jump and go over here, and notice now I have 2 sodium. Same exact thing, exchange reaction. And so what's happening is I am taking strong acids and converting them into weak acids and strong bases and converting them into weak bases, but I'm not getting rid of them. So they are quick, They function very rapidly, but they're a temporary solution. You can overwhelm these because we have to have the right ratio. We have to have a certain number of bicarb to acid for this to even work. And once we start running out of it or having too much, we're going to have acid-base imbalances. And when we look at bicarb, we're usually looking at something called metabolic. So this wraps up our chemical buffer lecture.